All right. Hello, everyone. Thank you so much for joining us today. It's wonderful to be able to introduce Dr. Lucio Miele from LSU. He is the Cancer Crusaders Professor and Chair in the Department of Genetics and the Assistant Dean for Translational Science at LSU Health here in New Orleans. He's also one of the co-directors of the Biomedical Information Core within the CaTS. So with all of the hats that you wear, Dr. Miele, thank you so much for talking to us today about a new druggable pathway for cancer immunotherapy, selective pharmacological modulation of Notch1 protein levels in CD8 T-cells. We are all being recorded just to let everybody know. And Dr. Miele, would you like to take it away?
Thank you so much, Megan, and thank you all for being here. This is rare these days that I talk about my laboratory research, the arm of my work that takes place in the lab. But what I have been interested in over the past 25 years is tapping Notch signaling for therapeutic purposes in cancer. What we're going to be talking about today is immunotherapy. Now, you know that by now, cancer immunotherapy is mainstream. It has been mainstream for about 10 years now. Which doesn't mean we've solved every problem, but we do have another tool in our arsenal. There are many kinds of cancer immunotherapy agents approved by the FDA, including cytokines, tumor-targeted monoclonal antibodies, as well as antibody-drug conjugates, checkpoint inhibitory monoclonal antibodies that have been the whole rage largely since 2018. TVAC is a virotherapeutic herpes virus derivative, as well as adoptive cell therapy with CAR T-cells and other kinds of cellular therapies. Now, all this sounds fantastic. However, responses to immunotherapy are still highly variable. Different tumors respond very differently. 25 to 30% response rates are generally observed. Just as is the case for chemotherapy and targeted agents, primary or acquired resistance occurs. And responses are affected by a number of factors. Some pertaining directly to the tumor, for example, how immunogenic is the tumor? With tumors containing a larger mutational load, often from defects in mismatch repair, being more immunogenic, but also the age of the patient and their general status, the evolution of clones inside the tumor with immune suppressive capacity, this is called immune editing, and multiple immune suppressive mechanisms in the tumor microenvironment. This is another way of showing the same data, essentially. The number of tumor-infiltrating lymphocytes appear, systemic comorbidities, environmental factors like diet, age, background infections, smoking, etc., etc. One important thing that has been recognized in several tumor sites is that not all tumors are immunologically hot, meaning they contain abundant T-cell infiltrates. The ones that do are much more likely to respond to immunotherapy. And for some of them, the situation isn't necessarily due to immune suppression, but to the actual structure of the tumor. For example, tumors very rich in fibrous tissue, like pancreatic cancers, are more difficult to infiltrate for T-cells. This is what I just told you, immune editing. It is thought, it's been suggested that transformed cells are eliminated all the time by adaptive immunity and also by innate immunity during the body. You can see it on the left, elimination phase. And this essentially sets up a quasi-Darwinian selection pressure to reach an equilibrium state where the tumor is dormant and it acquires characteristics that make it more and more stealthy towards the immune system, including the production of immune suppressant mediators. And finally, immune escape, where the tumor overcomes immune selection and it becomes macroscopic. In that case, it actually, there are features of the immune system that can actually promote tumor growth, such as myeloid-derived suppressor cells, Treg T cells, exhausted T cells that express checkpoints like CTLA4 and PD-1, as well as cytokines and other mediators. Now, we're going to focus today on one of the many immune suppressive mechanisms that tumors produce, and that is adenosine. Adenosine is a nucleoside. For those of you who remember your biochemistry, it consists of adenine and ribose. And it binds to a series of purinergic receptors called A2A, A2B, A3, and A1 that exist in numerous cells. They exist, as you can see here, the ones that I have labeled with red circles. These receptors are present in CD8 T-cells, in the tumor cells themselves, in Tregs, in blood vessels, and markers for high production of adenosine, such as expression of its receptors, but also of the enzymes that produce it, CD73 and CD39, portend to poor prognosis and high likelihood of tumor progression. We don't have time to go into all the details of how adenosine is produced. I can tell you the basics. It is a degradation product of extracellular ATP, which is produced in a hypoxic environment. ATP becomes ADP, AMP, and then adenosine. The primary generator of adenosine is CD73, which is an enzyme that catalyzes the bidirectional transformation of adenosine into AMP in the extracellular space. This is a membrane enzyme. But there's also, kinase and CD38. Now, once adenosine is released in the extracellular space in a hypoxic environment, it binds to its receptors. These are GPCRs, meaning G protein linked transmembrane receptors. As I said, we have A1, A2, A2B, and A3. And these have multiple different immunological effects on several cells of the immune system, suppressing T-cell activation, suppressing antigen-presenting cells, and stimulating myeloid-derived suppressor cells, as well as promoting the release of immune suppressive mechanisms by tumor-associated macrophages, which take on an M2 phenotype. In the tumor microenvironment, we find primarily A2A and A2B receptors in several cells of the tumor microenvironment, particularly in endothelial cells, immune cells, as well as cancer-associated fibroblasts. Also, exosomes that produce adenosine, because they have CD39 and CD73 on them, can be released by cancer cells, expanding the production of adenosine in the extracellular milieu. Expression of CD39, CD73, the A2A and A2B receptor correlates with poor prognosis in a number of malignancies. You can see here, going from ovarian cancer to triple-negative breast cancer, lung cancer, prostate, oropharyngeal carcinoma, non-small cell lung, etc. So this is a field where there's a lot of attention. And there are inhibitors of this pathway in clinical development, except they don't work very well because there are many different ways of producing adenosine. So, so far, the agents that have been tested to inhibit adenosine in the clinic have been somewhat disappointing. So why and how did I become interested in adenosine? This is a connection with the Salerno Medical School in Italy. I'm giving it its Latin name here because it is the oldest medical school in the world. It was founded in year 900 AD. It then eventually had a side campus, which became my alma mater, the University of Naples, in 1224. I go there once a year and I give CME events. And there I met a then junior faculty, Silvana Morello, who was an assistant professor, PharmD PhD in the School of Pharmacy, and her graduate student, Claudia Sorrentino. Claudia wanted to do her dissertation work in my lab. And so we arranged for her to come to New Orleans and work on a project. Now, of course, she came from an adenosine lab. And we've never worked on adenosine, we work on Notch. Not to worry, Spiros Artavanis Tsakonas, who is the scientist who isolated Drosophila Notch, and also my good friend, has an aphorism essentially indicating that everybody works on Notch. Some of us just haven't realized it. In other words, Notch is involved in so many processes that the question is not whether or not Notch is involved, but how. So I gave a challenge to Claudia. Does adenosine crosstalk with Notch? This is what we knew. We've known for a long time that through work of several groups, including our collaborator, Barbara Osborne, and ourselves, that Notch1 is required for CD4 and especially for CD8 T-cell activation. We also know that Notch1 is rapidly activated by the T-cell receptor upon antigen binding through a unique ligand-independent process. We also know from work that Paulo Rodriguez published when he was here at LSU that CD8 T-cells transgenically expressing the intracellular portion of Notch1 and ICD1 are super tumor killers, and they are resistant to several immune suppressive mechanisms. We also knew that CD8 cells express both A2A and A2B adenosine receptors. So we did some basic pharmacology, and we came up with the answer that I'm going to tell you in a second. I'm going to give you a really quick reminder of Notch signaling. We don't have time to go into details, but notches, there are 4 mammalian notches, are large heterodimeric transmembrane receptors that are primarily involved in close contact cell-cell communications, paracrine communications. They consist of an extracellular region that's non-covalently bound to a transmembrane domain. And there are 5 ligands, of which 4 are stimulatory and 1 is inhibitory. Jagged 1 and 2, homologous to Drosophila serrate, and delta 1 and 4, homologous to Drosophila delta, are stimulatory, and delta 3 is inhibitory. This is a complicated diagram of what happens when a ligand binds to Notch, but to make the long story short, it pulls out the extracellular subunit through actual mechanical force into the ligand expressing cell, leaving this, which is the transmembrane subunit, which is cleaved by an ADAM protease, releasing creating an extracellular intermediate, which is then cleaved by gamma secretase. Gamma secretase produces an intracellular fragment. This can be produced either at the membrane or in endosomes. And there are many, many mechanisms, keep this in mind, that control the total amount of Notch protein at the cell membrane, because Notch signals are extremely, extremely dose dependent. Even a 50% difference in the amount of Notch makes a dramatic difference in the phenotype. Once the intracellular region of Notch is produced, it does two things. It signals in the cytoplasm to a variety of non-nuclear signals. And in the nucleus, it becomes a transcriptional regulator, replacing a co-repressor complex with a co-activator complex on a transcription factor called CSL or RBPJ kappa, thereby promoting the transcription of Notch target genes. Some of which, for example, in CD8 T-cells include gamma interferon, granzyme, and perforin, which are necessary for cytotoxic T-cell activity. Non-canonical Notch signaling, we don't have time to go into for this purpose. In T-cells, things are a little different. And this was shown by Barbara Osborne's lab. Notch is activated in a ligand-independent fashion. So if you take a T-cell and you give it its antigen and a co-stimulatory signal, you're going to get Notch activation whether or not there are ligands around. And this is due to endocytosis, which is promoted by PI3 kinase, PKC theta, and ADAM17, which cleaves Notch here. The acidic environment in the endosome unfolds the extracellular subunit, and you get ligand-independent activation of Notch, which is necessary for T-cell activity. So what did Claudia find out? That adenosine downregulates Notch1 protein, but not mRNA in CD8 T-cells. That this effect is mediated by the A2A receptor, that knockout cells for the adenosine receptor do not downregulate Notch1 in response to adenosine, and that Notch1 and intracellular portion transgenic T cells are insensitive to adenosine, essentially. And this was published a couple of years ago. Enter Giulia Monticone, my postdoc, who joined me from Manchester, where she got her PhD in non-canonical Notch signaling. So I said, well, Giulia, Claudia graduated. We have a bunch of open questions still on this business of Notch in T cells. And these are some of the questions. How does A2A modulate Notch protein? Does protecting Notch1 from degradation actually enhance anti-tumor CD8 responses? And can this be leveraged pharmacologically? Is there a selective druggable target that enhances the stability of Notch in T cells, but not in cancer cells? We don't have time to go into that, but a number of tumors use Notch as an oncogene or as a tumor promoter. So if we're going to stimulate Notch, we need to be absolutely sure that we only do it in T cells and not in the tumor cells. So these are some of Giulia's answers. The antagonist ZM, a selective antagonist of the A2A receptor, does in fact enhance the anti-tumor activity of Notch1 positive CD8 T cells. You can see this in organoids, both in phase contrast and in immune fluorescence, where green is dead cells, red is total cells, and blue is DNA. And you can see that with ZM, there are many more dead cells in tumors, provided, this is important, that these organoids come from immune competent animals. This is the line above. In immune deficient animals, there is no effect whatsoever, indicating that this is not a direct effect on the tumor, it is an immune effect. You can see here what happens to the growth of organoids. CGS is an agonist of the receptor, thereby expected to suppress immune responses, and it does. And ZM completely wipes out organoid growth, and it competes with CGS. This effect is dose dependent, and as you can see here in organoids from immune-compromised mice, there is no effect. There was also an increase in Notch1 positive CD8 T cells in the organoids, as you can see here pictorially and here statistically. Okay, we're on the right track. So we knew that the effect of adenosine was at the level of protein rather than mRNA. What we found out is that adenosine modulates the ubiquitination of Notch1. You see here protein levels of the full-length notch and the transmembrane subunit and the intracellular subunit. CGS, which is the agonist of the receptor, causes notch degradation. ZM does not, and it prevents the effect of CGS. What CGS does is it greatly increases the amount of ubiquitinated Notch1. And it decreases the half-life of the protein in cycloheximide experiments, as you can see here. It also inhibits the proliferation of CD8 T-cells stimulated with anti-CD3 and anti-CD28, and essentially abrogates gamma interferon production. Enter our collaborator, Barbara Osborne, who had been working on the ubiquitination of Notch1 independently. And this is data from Ameya Chandrasekhar in her laboratory. The ubiquitin ligases, the ubiquitinate Notch, and there are several, include in T cells primarily Cbl-b. Ameya co-immunoprecipitated Notch1 with Cbl-b and also mapped the interaction. We don't have time to go into these deletion mutant experiments, but she essentially identified the region of Notch and the region of Cbl-b that interact with one another. And importantly, when you transfect cells with Cbl-b, you decrease the transcriptional activity of Notch. This is in HEC293 cells, so even outside of T cells. A knockout of Cbl-b, a somatic knockout of Cbl-b, as you can see here, there's no Cbl-b anymore, results in higher levels of Notch1 transmembrane proteins in both CD4 and CD8 T cells. Okay, but how does Cbl-b find Notch1? This required some proteomics, which was performed at the University of Arkansas, thanks to a LACATS voucher, so thank you. And essentially, we looked at the Notch interactome with or without ZM versus CGS. In other words, agonist versus antagonist. And as you can see here, there were a few proteins that were prominently modulated by engagement of the adenosine receptor, of which the most modulated one was STS1. Well, it turns out that STS1 is a Cbl-b adapter. So confirming Barbara's data, the interaction between STS1 and Notch1 was modulated by ZM and CGS, as you can see here. Now, STS1 is a phosphatase which modulates the phosphorylation of Cbl-b, as you can see here with phosphotyrosine immune precipitation, and cells from animals that are knockout for STS1 and STS2 are insensitive to CGS. All right, so we have placed another component into the pathway, which is STS1 and Cbl-b. Okay, but that still doesn't tell me what do I do with this pharmacologically. Remember, I'm a translational guy, so I want to know what do I do with it. So we identified a company in Cambridge, Massachusetts, Nimbus Pharmaceuticals, which is developing Cbl-b inhibitors for immunotherapy. So we contacted them and we said, look, we may have found a possible mechanism of action for your compounds. Are you interested in collaborating? And so the answer was yes. And they gave us their 2 lead compounds. And to make the long story short, the Cbl-b inhibitors protect Notch full length and transmembrane and intracellular from degradation induced by CGS, which is the A2A antagonist. They stimulate T-cell proliferation and rescue the effect of CGS on T-cell proliferation, on gamma interferon, and on granzyme. So this is further proof now pharmacological as opposed to genetic that Cbl-b mediates the effects of adenosine on Notch, and we may have a small molecule way of dragging this pathway. Well, do these molecules work? Now, the company at the time was still deciding which of these molecules had the most favorable pharmacokinetics in vivo. And so we did these experiments in organoids. First of all, we showed that, yes, indeed, Cbl-b inhibitors do increase the number of Notch1 positive CD8 T-cells in organoids and organoid killing. Once again, in an immune-dependent fashion, because organoids coming from immune-suppressed animals do not respond. The number of Notch1 positive T-cells in organoids was increased upon treatment with their most potent lead compound. And we tested this in 3 different models. The CO321 model is an interesting one because it is Notch dependent. It's a triple negative model that is deficient in lunatic fringe, which makes it Notch dependent. This was important because we did not want, remember, to stimulate Notch1 in the tumors. The short answer is we don't, and we still have a strong immune therapeutic effect. The M-Wnt model, which is driven by Wnt, also triple negative, and colon cancer MC38 models. So this effect does not appear to be model specific. So what is Julia's response? That A2A receptor triggers Notch1 degradation through a novel pathway that involves STS1 and Cbl-b. Cbl-b ubiquitinates Notch1 and causes it to be degraded in response to adenosine. A CRISPR-Cas somatic knockout of Cbl-b and investigational Cbl-b inhibitors rescue Notch from degradation. And these inhibitors enhance the anti-tumor activity of CD8 T-cells in tumor-derived organisms. I haven't shown you that they also greatly enhance the efficacy of anti-PD-1 and anti-PD-L1. These results were published 2 weeks ago. This is the working model so far. T-cell receptor through LCK would cause the phosphorylation of Cbl-b, which makes it fall off STS1, adenosine through A2A, and PKA, and most likely CSK kinases, causes both the dephosphorylation of LCK and the dephosphorylation of STS1, which now sticks to Cbl-b and adapts it to Notch, causing it to be degraded. By eliminating this arm of the pathway, we're preserving more Notch upon T-cell activation and enhancing T-cell functions. Do we still have more questions? Of course we do. Are other Notch paralogs regulated in the same fashion in T-cells? We know that Notch2 most likely is. We need to look at Notch3. What about NK cells? Spoiler alert, the answer is most likely yes, and this is very important for adoptive cell therapy. Do other cyclic AMP upregulate immune suppressive signals, such as prostaglandins, modulate Notch1 levels in the same fashion? Do immune checkpoint inhibitors interfere? Do they modulate Notch? And what is the in vivo safety and efficacy of the lead Cbl-b inhibitor? This is in progress. And can one make super CAR-T or CAR-NK cells with loss of function of Cbl-b, as opposed to expression of an ICD, which is an oncogene? This has been a very quick rush through this project. As I said, the 3 colleagues who are primarily responsible for this data are Claudia Sorrentino, Giulia Monticone, and Ji Huang. but also Fokro Hossein, Antonio Pannuti, Sam Majlander, Denise Ukar, Julie Crabtree, and Sierra Singleton. And of course, our multiple collaborators, Barbara Osborne and Lisa Minter at UMass, Todd Goldie at Emory, Silvana Morello at University of Salerno, Paolo Rodriguez and Moffitt, Misha Sitkowski and Stephen Hatfield at Northeastern, who gave us the age-way knockouts, Christine Lowe and David Ciccone of Nimbus, who gave us the inhibitors, and Nicola Scarpino, who gave us the STS1 knockout mice. And I'm going to, happy to be taking any questions.
Excellent. Thank you so much, Dr. Miele. That was a phenomenal talk. If anyone has questions, they are welcome to unmute themselves or raise their hand, I guess, and ask. I'll unmute you if you'd like, or if you'd like to type it in the chat, we can have questions that way as well.
Keep in mind, I have to rejoin my other conference in a few minutes. So
we'll make it quick as possible.
Yeah.
Okay, hearing none, seeing no hands raised, seeing no chat. Dr. Miele, you're off the hook for the questions. But thank you everyone for joining us for today's CaTS seminar series with Dr. Miele. We greatly appreciate it. It was recorded, so it'll be available in the next day or so on the CaTS YouTube channel. And please plan to join us in October for Justin Brown from Pennington Biomedical Research Center talk. We'll be doing it again on October 20th at 12:30 p.m. We look forward to seeing you all then.
Thank you, Megan, and thank you, everyone.
Thank you so much. Have a great rest of your day.